Okay, so let's talk about the central dogma of molecular biology. Okay, so basically this is just about the process of replication, transcription, and translation of our genetic materials into something useful. Okay, so before we describe yung nucleic acids, no? So nucleic acids ay polymer similar with proteins, di ba? And meron siya mga bases, no? Which are called nucleotides, no? Ang monomer ng proteins ay amino acids. Ang monomer ng uh, nucleic acid naman ay nucleotides. No? Nire-recall ko kung nagkabaliktad ba ako ng sinabi. Basta yan. Pag proteins, amino acids, pag nucleic acid ay nucleotide. And our nucleotides are composed of three parts, di ba? Tatlong parts yun. You have the sugar, which could be deoxyribose or ribose. You have your phosphate groups, no, which will serve as our backbone. And then we have our nitrogenous bases, no, na purine and pyridines, no. So apat yung combination natin na yon, including one for RNA exclusively. Okay, so we have the adenine paired with thymine or uracil, and then we have the cytosine paired with guanine. Okay, so adenine and thymine or uracil are paired with two hydrogen, no? Hydrogen bonds yung kaya nila, dalawa. But for cytosine and guanine, tatlo yung hydrogen bonds na kaya nila. And we also discussed last time that the ano, the strand of our nucleic acids, no? So, yung backbone nun is the polydiester bond, di ba? Polyphosphodiester bonds, which runs from 5' to 3'. And then the complementary strand, no, yung kapartner niya sa kabila, runs from 3' to 5'. Okay? So basically, they are anti-parallel in direction. Okay? So, kaya ang itsura niyan ay alpha helix kasi anti-parallel yung kanyang direction. Okay? What else, no? Meaning ng complementary ay kung adenine yung nasa isang side, yung kapartner nun yung nasa other side, no? So, ang kapartner ni adenine is the thymine and si guanine ay sa kabila ay si cytosine and vice versa. Okay, so ibig sabihin, sila-sila lang din yung magkaka-pair, no? So, they complement each other. So, suppose, kunwari sa isang strand ng DNA, sinabi ko doon, meron nun sa quiz nyo, sa isang strand ng DNA nakalagay ay CCC AAA. Okay. Ang complementary nun would be the GGG TTT. No? So yun yung kanyang complementary. But suppose we have a strand that is ATCG. Okay. Ang complementary nun ay TAGC. Okay. So ganun yung kanilang mga strand. No? So they are complementary to each other, anti-parallel, and they form the alpha helix. So yung mga idea na yon, gagamitin natin yon later on to briefly describe what happens in the central dogma of molecular biology. No? Okay. So the overall summary for the flow of genetic material or genetic information in cells, whether that's prokaryotic or eukaryotic, is this. No? So you begin with your DNA, okay? So when your cell is duplicating, replicating, so nauulit-ulit yung DNA, dumadami-dami yan. No? So discuss natin mamaya yung process ng DNA replication. But if the cell requires to produce some proteins, pag gusto niya gumawa ng protein, gagawin niya is that yung DNA niya ay i-convert niya partly to RNA. No? A portion of the DNA will be coded to form the RNA. And yung RNA, mare-replicate din yan, no? Similar with the DNA. Okay. And then, this RNA, no? So ito, nasa nucleus to. Kung gusto na ng cell natin gumawa ng proteins, lalabas na siya. Yung RNA, lalabas na siya sa nucleus, papunta sa ribosome. Wherein, in the ribosome, the RNAs will be translated into usable biomolecules called proteins. Okay. If we do the reverse of RNA to DNA, tawag doon ay reverse transcription, which is familiar tayo. You know the RT-PCR? Yung RT doon means reverse transcription. No? So basically, from the genetic material of the virus, pwede mo makuha yung DNA. No? Pwede mo malaman kung COVID positive siya or not. No? So, dinedetermine ng RT-PCR if you have presence of viral RNA materials. No? So, discuss natin yan very broadly later. Hindi na natin masyado lalaliman, no? Kasi masyadong malalim tong topics na to. Pero what we want to get is just the overall knowledge lang. Okay. So, yan. So, let's briefly describe each processes, no? So, for DNA replication, it is defined as the copying of the parent DNA to make daughter DNA, no? So basically, yung DNA mo na isa, paparamihin mo. Okay? And that is important. Especially, our cells are known to be dividing always. No? Every now and then, our cells divide. No? Para ma-replenish yung mga namamatay din na cells. No? Okay? So, in order to preserve our body, para ma-preserve yung ating katawan. No? So, the DNA has to replicate, no? Para dumami din yung cells natin. Okay? And then the next process, if you want to make proteins out of the DNA, no? Okay. Gagawin natin yung transcription going to translation. So, nangyayari sa transcription is that sa ating DNA strand, no, portions of it will be coded, no? Maliliit na portions lang i-convert into RNA. Parang ifo-photocopy lang niya yung ilang pages, no? Kasi yun, transcribed na siya. Pag translation naman, ito na yung conversion of the RNA into something useful. That is the protein. Okay. So imagine nyo itong process ng central dogma as parang pagtuturo din. No? Kasi kayo, magiging teacher din kayo in the future. So imagine nyo, yung DNA replication, o yun yung yun yung mga libro-libro na kinukuha natin. Yung transcription, only portion of that will be read, no? Every now and then. And then yung translation, yung ikaw na yung magtuturo sa students. No? So from the book, kukuha ka ng certain chapter, ta-translate mo siya into something na understandable. No? So parang pagtuturo lang din to. Okay? Although this one is much more complicated than teaching. Okay? Mahirap talaga tong topic na to. Kaya light lang natin siya i-approach. Ha? Okay. So special cases is of course the reverse transcription or reverse transcriptase, no? Yan yung pagbaliktad ng process wherein we know yung DNA magiging RNA yan, no? Along the process, no? Pero in reverse transcription, what we're going to do is that from RNA, we are going to convert it to DNA. So baliktad, no? So this technology is used in determining viral materials in our body, especially for HIV testing. Okay, so gumagamit doon ng RT-PCR din for HIV. Ginagamit din to for coronavirus, no? COVID-19. Okay. So ginagawa lang doon is that it will detect, no, from the cell material natin, from our blood or from our mga fluid sa katawan, yung mga kinukuha sa ilong, no. So kukunin doon yung mga genetic material, and then whenever they found out that andito yung RNA material na to, yun. So masasabi natin whether you are positive or negative sa viruses, no. Okay? And this is this technology is widely used ngayon, especially pandemic. Pero even before, no, gamit na gamit tong RT process na yun, no. Kasi they are trying to create molecular advances, no, in treating viruses, no, infections such as HIV, no. They are actively seeking for its cure kasi until now, no. Even cancer, no, ginagamitan din nila ng RT methods. Okay. So ayan. So this is currently undergoing improvements, no? And with the help of our new technology, the mRNA vaccines, no, mas bumibilis yung ating production ng cure sa diseases, no. And as of last time, no, may nabakunahan na for AIDS, no. May nabakunahan na for HIV virus, no. Yan. So hopefully maging successful 'yon para hindi lang COVID yung mawawala, even that HIV na. Okay? So marami rin kasing nadadali ng virus na 'yan eh. Okay? Maraming buhay na rin ang nasayang. Okay? So 'yan. Okay, so this is the idea. Ang alam natin, the usual flow of genetic information is from DNA, magiging RNA yon, then magiging protein. No? But in RT-PCR, baliktad gagawin mo. No? From the RNA, ko-convert niya yon into DNA. No? So pag translate na yon into DNA, na-detect niya, uy, katumbas to ng sa COVID-19 disease. So, papasok kasi sa genetic material mo yung disease na yun eh. So, yun. Ayan, masasabi niya kung positive ka. Okay? So, yan. So, anyway, so going back to our main flow, ito yung main flow natin, yung yellow. Okay? So, isa-isahin na natin yung flow. Okay? So, let's start with the DNA replication. Okay? So, in DNA replication, it is said to be somehow semi-conservative in process. No? When you say semi-conservative, no, it retains half of its original traits. No? Kaya nga, in biology, alam nyo na yung offspring, yung mga anak, no, yung traits nila ay nakukuha nila from their parents. And yung parents nila, yung traits nila nakukuha nila from their parents din. No? And so on and so forth. No? So, bakit ganun, no? Kasi ito yung sabi ng biology, ito yung sabi ng biochemistry, that DNA replication is a semi-conservative process, no? Or semi-conservative in fashion. 50-50 yung 50-50 yung magiging daughter DNA natin, no? Okay? So, kunwari, you have your genetic material from your mother, genetic material from your father. Pag samahin mo yon, you have your offspring. No? Okay. So, yan. And yung traits nila ay evenly divided. Okay. So, the experiment that was used to confirm this semi-conservative property of DNA replication was the experiment by Meselson and Stahl. Okay. So, in their experiment, they use E. coli, no? E. coli yung madalas gamitin dito. Number 1 kasi ang dami na to sa paligid. Nasa tae lang yan. Literal, no? Pasensya na kung kumakain kayo. Pero yun, this is very abundant everywhere, no? So it is abundant in especially fecal matter. So madali siya i-collect, no? Madali siya i-harvest. So yun. So in the experiment of Meselson and Stahl, they analyzed the DNA replication process in E. coli, in prokaryotes. So yun, nakita nila that when they did genetic transcription, kinonvert nila yung mga sequences ng kanyang nucleic acids, napansin nila na 50-50 yung hatian ng kanilang materials. Okay? So 50-50. 50 from the other, 50 from the 50 from the mother, 50 from the father. Parang ganun yung idea din dito. Okay? So yan. So pero dito walang mother and father dito, no? So ang nangyayari dito is the parent cell lang, yun yung mag-split into 2. Okay? Pero alam natin in humans, kailangan dalawa, no? Kailangan dalawang genetic material. Okay? So, but the overall idea is that 50-50 ang split ng genetic information. So, yun yung reason kung bakit natin tinatawag siyang semi-conservative. Okay? So, anyway, so going back to replication process, di ba alam natin that our DNA is double-stranded yan. No? So, one strand will go from 5' to 3'. The other strand will go from 3' to 5'. No? So, paano mag-start tayo ng replication? Ganito. So, doon sa ating DNA strand, no? 'Di ba? Chromosome, nakapulupot lang yun na DNA. So, mag-unwind yun eh. Okay. So, mangyari doon is that there will be a specific point in your DNA helix, no? So, sa isang mahabang helix na yan, there will be a specific point wherein the replication will happen. And that point is called the origin of replication. Okay? So, dun lang talaga siya nangyayari. Okay? Sa origin of replication. Okay? So, yan. So, once na ma-identify na yung ating origin of replication in our DNA, no, medyo mahirap to mahirap siya i-identify exactly at experimental procedures kasi nga observe lang natin is yung products eh. Okay. Pero sinasabi nila there's a certain point kung saan mag-start lahat, no. Parang universe lang. Sabi nila mag-start yan at singularity. Ganun din sa DNA. It will start at the origin of replication. Where is that? We don't know. Okay. Basta doon siya mag-start. Okay. So once we have the origin of replication, ano mangyayari? Mag-unwind na yung ating DNA. Doon na siya mag-start mag-unzip, and then the replication process will be synthesized from 5' to 3' direction. Okay? So the creation of the new DNA will start from 5' to 3' direction. Okay? So again, ano yung idea lang dito sa slide natin na to? Hahanapin niya from your genetic material, hahanapin niya yung origin of replication. What is that exactly? We're not really sure, pero dun siya mag-start. Okay. So once the origin of replication was identified, then your DNA helicase, your DNA double strand will start to unzip, no, with the help of an enzyme. Okay. So pag na-unzip na yon, doon na mag-start yung replication in 5' to 3' direction. Okay? So, yan. So, yan. Sabi dyan, no? All synthesis of nucleotide chains occur from 5' to 3' direction. Okay? So, yan. So, hindi na natin babasahin yung too much details, ha? Kasi too complicated na yung iba dito. Okay. So, anyway, this is the overall process in the replication process. No? So, replication pa lang tayo, ang dami nang nangyayari. Okay? So, ito yung steps. No? So, syempre, hahanapin mo muna yung origin of replication. Once it is identified, then you will start doing the replication. Ang first step done is the initiation process. In the initiation process, dito na mangyayari yung unwinding of the DNA. So parang ito yung sa zipper, di ba yung zipper? Imagine nyo DNA yan. Doon na mag-start siya ma-unzip, no? Mahahati na siya into 2. Okay. So ganun din DNA natin. So our DNA, okay, mag-split siya into 2. Okay. So with the help of DNA helicase, doon siya na-unzip. Okay. And then to help minimize the stress, no, when you are unzipping the DNA, we need to use an enzyme called the topoisomerases. No? So they relieve the stress when unwinding DNA. Okay. So try nyo yan, 'di ba? Dati may headset tayo. Nung buhay pa yung CDR King, wala na kasi ngayon eh. Ginagawa ko, yung headset kasi doon, yun eh, 2 pair yun, 'di ba? 2 lines. Ginagawa ko binabata ko yun ng bata ako. Kaya sira eh. So, pagkabata ko noon, minsan nabubuhol siya, no, bago dumating sa dulo. So, ganun din kasi DNA, no. Kapag binigla mo yung pag-unzip, pwedeng masira, no, mabubuhol yung DNA mo. So, to minimize that stress in uncoiling your DNA, we need topoisomerase, no. Okay, so yun yung mag-guide, no, to make the unzipping better, no. Parang siya yung zipper sa zipper. Yan, so yun lang yung idea. And then once our DNA is split na, it will now undergo the process called elongation. Okay. So in the elongation, ang daming nangyayari. Okay. Bakit? Kasi remember, dalawa yung strand natin. We have a strand that goes from 5' to 3', and then we have the other strand that goes from 3' to 5'. Dalawa yun, di ba? So yung iba yung process for 5' to 3', iba rin yung process for 3' to 5'. Okay? Kaya sabi ko nga, yung general idea na lang kukunin natin dito. Okay? So, yan. So, we have two separate processes for each strand, no? For each parent strand. Okay? So, may mga enzymes na gagamitin doon to help the process, no? So, minsan gagamit tayo ng primers, which are RNA molecules or nucleotide bases, no? Minsan gagamit tayo ng DNA polymerase 1, 2, 3 to help synthesize the DNA, and then we need Okazaki fragments. So, mamaya discuss natin yan later. So, yan. So, iba yung process for 5' to 3', iba yung process for 3' to 5'. And then after doing that, no, once our parent strand are paired with their complementary bases na, the new complementary base, matuterminate na yung replication process na. So you started with just one pair, no? 5' to 3', 3' to 5'. Pag nag-split yan, papartneran sila ng panibago nilang kapartner, no? Papartneran sila ng panibagong strand, no? Okay. And then once complete na yung replication process, then mataterminate na yung ating DNA replication. May dalawa na tayong DNA. Okay? So to investigate this, no, kasi kapag mag-start ka agad with eukaryotes, which is animal cell, medyo complicated, no? Kaya typically, when it comes to biological processes, they really start with bacteria. So yung bacteria yung inaaral nila. Bakit? Kasi the cellular structure of bacteria are simpler than eukaryotic cell, di ba? So prokaryotes mas madali aralin kasi maliliit lang, not too complicated unlike animal cells, no, that we have. So yan. So when they studied the DNA replication in prokaryotes, they found that na similar din yon sa eukaryotes. Okay. So ito yung nangyayari, ito yung summary. Although ito yung pinaka-summary, ito yung much detailed summary. Okay. Papansin nyo, ang dami nangyari, no? So, ano mangyayari dyan? Your DNA, it will unwind the origin of replication. Then, the helicase will open yan. I-unzip nyo yan. With the help of topoisomerase, malilesen yung stress. No? And then, to prevent the DNA from sticking back together, no? Kasi madikit yan eh. May mga hydrogen bonds yan eh. So, to prevent that from sticking together, kakabit yung ating mga SSBs or the single-strand binding proteins. No? So, these are proteins that prevent the DNA from going back together. No? Para continuous yung replication process natin. Okay? And then we will have primases, no? So these are enzymes that will help to attach the RNA primers complementing the DNA strand, no? And then gagawin yung DNA polymerase, papalitan niya yung mga RNA into DNA nucleotides. So yung U gagawin na niyang T. Okay? Then elongation, tutuloy-tuloy yan until later on, tapos na yung process. Okay? So, ito yung idea lang. Okay, so we have this double helix here. We have this strand from 5' to 3'. Then we have 3' to 5'. Okay? So, yan. So, this is your topoisomerase. It will help relieve the stress here. And then this is your helicase. It will unzip the hydrogen bond here. And as I told you earlier, yung SSBs natin, this will prevent them from coming back together. Kasi baka mamaya, bumalik lang sila doon. So to prevent them from sticking together, yan. So kakabit sila doon sa in-between ng DNA strand natin. Okay? So there is a, since ang ating DNA na-split na siya into two, sabi ko nga sa inyo, meron tayong, 3' to 5', and then we have the 5' to 3'. No? So magkakaiba yung steps dito sa pag 5' to 3' at sa 3' to 5'. Let me denote na. It's the 3' to 5', 5' to 3'. Sa 5' to 3', ang tawag natin doon ay leading strand, no? Okay. Kasi sabi natin, the transcription goes from 5' to 3'. I mean, the replication pala, sorry. So the replication goes normally in 5' to 3' direction. Okay. So since itong ating DNA strand na to is 5' to 3' yung strand na yan, so continuous yan, no? Tuloy-tuloy yan. Kaya leading strand tawag dyan. No? Tuloy-tuloy lang yung process niya. Wala masyadong nangyayari dito. Ang nangyayari lang is nadadagdagan lang ng nucleotide bases itong ating strand na to. So we will have a new DNA. Ang problema is kapag 3' to 5' yung strand mo. Okay? Pag 3' to 5' yung strand mo, may ibang technique silang ginagawa dito. Okay. Ang ginagawa ng ating cell is ganito. 'Di ba sabi ko nga sa inyo, pag 5' to 3', dagdag nucleotides ka lang. Pero kapag 3' to 5' iba. Gagawin niya is ganito. Chop-chop niya yung gagawin, no? So kunwari, itong portion muna gagawin niya up to here. Tapos itong portion up to here, no? It will try to do the replication in 5' to 3' process, no? So kumbaga, i-chop-chop niya pa yan para mag 5' to 3 5' to 3' yung kanyang process ng pag-add ng nucleotides. So kunwari, dito muna siya. Tapos after niyan, dito naman ako. Tapos after nun, dito naman siya. So bale, china-chop-chop niya itong lagging strand natin. China-chop-chop niya so it can work in 3', in 5' to 3' direction. Opposite yung direction niya eh. So dahan-dahan na. Sabi niya, wait lang, ang dami eh. Dahan-dahan muna ako. Kasi kailangan niya mag 5' to 3' direction. Okay. Kaya tinawag natin yung lagging strand, no? Lagging strand because baliktad yung process, no? So against the flow siya eh. Ito yung flow eh, 5' to 3'. Kaya nga sa leading strand tuloy-tuloy lang. But in the 3' to 5' strand, against the flow yung direction ng reaction. So, putol-putol. As a result, lagging strand yan. So dito nagkakaroon ng kakaibang process kasi there are some enzymes that are special for this process, no? Okay. So ang gagawin niya is ganito. Si DNA ligase, i-identify niya, saan ako mag-start, no? Sabi niya, dito ka mag-start. Ayan. So pag sinabi niya ni DNA ligase, dito ka mag-start mag 5' to 3', opposite direction. Then saka papasok si DNA pol 1. Okay. So si DNA polymerase 1, siya ay magdadagdag ng nucleotide pair for the lagging strand in the 5' to 3' direction. Ang tawag dun sa pair na dinadagdag niya ay Okazaki fragments, no? Bakit fragment? Kasi nga patche-patche yung ginagawa niya, no? So chop-chop niya yan eh. Sabi ni ligase, dito ka muna. Add mo yung Okazaki fragments. So gagawin ni DNA pol, ilalagay niya yung Okazaki fragments. Tapos after nun, sasabihin ni DNA ligase, ito naman. Tapos doon, papasok ulit si DNA pol 1. Add mo dito yung Okazaki fragments. Doon naman siya. So, magko-continuous yun. Continuous process yun. So, si DNA ligase, sasabihin niya parate, dito ka mag-start. Dito ka mag-start. Dito ka mag-start. Si DNA pol naman, susunod lang siya. Si DNA pol 1, siya yung magdadagdag ng Okazaki fragments. Then, si DNA polymerase 2, siya yung nagpa-finalize niyan, no? For example, ito yung isang Okazaki fragment, ito yung Okazaki fragment. Yung space in between, siya na yung nagpa-finalize, no? Siya na yung nagtatapos ng gap nila. So, it adds the replacement or the final nucleotide base, no? Para makomplete yung ating lagging strand, no? Okay. So, medyo mahirap siya i-visualize, no? Pag pag sinabi ko lang in words, no? Kaya, ang gagawin natin is, manonood tayo ng video. Okay. Ano lang siya, no? Pag naalala mo na yung process, or pagkabisado mo yung process, medyo madali lang siya. Pero, kapag, ngayon pa lang natin to na-encounter, which is true naman, okay, medyo mahirap siya i-visualize, no? Okay, so to help us visualize this DNA replication process, let's watch this video. Let me know kung walang sound ha. Pero I think na-turn on ko na yung sound. So this is a short clip which will describe the process of replication. Makikita nyo dito na yung ating DNA, it will be split by the DNA helicase, and the topoisomerase will help the stress to be lessened. And then yung isang strand, the leading strand, is tuloy-tuloy lang yung addition ng nucleotides. But for the lagging strand, doon maraming helpers. Doon gagamit tayo ng DNA primase, doon tayo gagamit ng DNA pol 1, DNA pol 2, and doon kinakabit yung Okazaki fragments. Okay. So to help us visualize the process better, let's watch this video clip. Let me know kung may sound o wala, ha? Pag may sound, ayun, sabihin nyo lang.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence.
By the way, may narration. May naririnig ba kayo?
Yes po.
Meron naman?
Yes po.
Okay, sige. May narration. Basically, yung sinabi ko kanina, naka-English lang dito. Tapos may visualization para better. Okay?
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, di ba mas madali kapag may visuals? Pag face-to-face yan, ang hirap i-drawing yan. Thank you sa online natin. online platform, YouTube, no? So anyway, so visible naman yung steps na sinabi
DNA
ko sa inyo
point for the
kanina, no? So ano yung step na yon? Number one is that your DNA will be unwound, no? Ano siya, split siya ng ating helicase, no? May kasama ditong may kasama ditong topoisomerase, di lang pinakita, no? Okay. But for other YouTube videos, may isasama nila 'yan. Okay? Especially yung lengthy YouTube videos. Okay? So, mangyayari diyan, mag-split siya. Apparently, ang tawag sa splitting na 'yan ay replication fork, no? Share ko lang, no? 'Di ba, papansin nyo, yung ating mga gamot, no, especially multivitamins, may nakalagay doon zinc, no? May zinc na nakalagay doon. Bakit kaya, no? Bakit kaya may zinc sa ating multivitamins? And even yung doctors, no? If magpa-check up kayo, ang i-recommend nila sa inyong multivitamins are those with zinc, no? Yung iron kasi okay lang yun eh. Pero yung zinc important yun. Bakit kaya? Kasi ito yung sagot, no? In DNA replication, the replication fork, no? Itong fork na to, this is actually stabilized also by zinc, okay? So, ibig sabihin, if marami tayong zinc sa katawan, mas madali yung DNA replication process. And as a result, we will have much more healthy cells. No? We will have much more healthy cells. Kaya hindi tayo magkakasakit. No? If ever man magkasakit tayo, mabilis mare-replace yon ng healthy cells. No? Mag-undergo ng cell death yung yung affected, na mag-apoptosis siya. Tapos yung natira, yung mga healthy cells, magpaparami sila with the help of zinc, no? So that's why it is important for you na uminom ng multivitamins with zinc, no? Ganong ginagawa ko now. So ang multivitamins na recommended by doctors, again, this is not hindi ako paid to do this, no? Ang iniinom ko na maganda is the Immune Pro kasi may zinc 'yon. And dati kasi everyday ako umiinom nun, no? Kaso medyo mahal nga lang. Alam mo naman, pag namamahalan tayo, kaya bumili ng bagong cellphone, pero yung vitamins hindi kaya bilhin. Yan yung weird sa tao eh. No? Kaya bumili ng Chicken Joy, pero vitamins hindi makabili. No? Yun kasi, no? Before, nung face-to-face pa, 2018, 2019, 2020, bago mag-pandemic, umiinom talaga ako ng Immune Pro. Napapansin ko talaga sa akin is that if I drink multivitamins with zinc, hindi talaga ako nagkakasakit kahit puyat ako, kahit overwork ako, no? Imagine nyo, sa PUP ako noon, pasok ako ng 7:30 AM, ang uwi ko alas 9 ng gabi. Minsan kakain lang ako, ilang meals lang ako in one day, no? Kasi puro teaching ginagawa ko. Pero himala, never ako nagkasakit, no? Kung kailan ako nasa bahay, doon ako nagkasakit, no? Ayun. So, ayan. So, hindi kasi ako umiinom ng zinc lately, no? Ngayon, umiinom uli ako ng zinc. Nakaka-recover ako. Okay. So, yan. The zinc actually helps the replication fork to be stable. Side kwento ko lang yan, no? Pero ang gusto ko sa inyo is next time na iinom kayo ng vitamins, make sure it has zinc, no? Other than the essential vitamins that we need, no? Anyway, so proceed tayo. So sabi ko nga sa inyo, the DNA is split now into two. We have the 5' to 3' direction, and then on the bottom part, we have the 3' to 5' direction. So since the replication process naturally occurs at 5' to 3' direction, tuloy-tuloy yon for the leading strand, no? So yan. So we have the primer, dadagdag niya yung primers, then your DNA pol will add the nucleotide bases, and that will continue to happen. Okay? So tuloy-tuloy yan, no? And so it will be continuous process, no? So for the leading strand, tuloy-tuloy yan because the direction is with the flow of the unzipping, no? Ayan, nasa flow siya ng unzipping yun, 5' to 3'. addition of the strand. However, for the lagging strand, opposite to. This is 3 prime, this is 5 prime, no? I mean, this is 3 prime, this is 5 prime. Eh, pag ganun yung direction eh. Kumbaga, ito papunta sa, right. Ang direction ay papunta, wait lang, nalito ako. So, ang direction nito is, left to right, no? Tapos, ito dapat, mahirap pala sabihin left to right. Sabihin ko na lang, ito, this is 3 prime to 5 prime. Ang kailangan, 5 prime to 3 prime. So, baliktad yung direction. Pag ganun to, ang dapat pag ganun. So, gagawin dyan, fragment. Ito yung sinasabi kong patse, no? So, fragment yung addition ng nucleotide bases. So, still, you will have your primer, DNA primer, and then you will have your Okazaki fragments, no? So, idadagdag na sila slowly. Yan, dadagdag na sila. Okay? So, tuloy-tuloy yan, no? Pero patse, kaya nga laging strand yan, no? Siya yung delayed, no? Okay. So, fragment ang addition ng ating DNA doon. And that's called the Okazaki fragments. Okay. And then for the termination step, yun, pinakita sa inyo kanina, the exonuclease, no? They remove the primers, no? And replace it with, they remove the primers and replace it with the final, nucleotide base, no? The appropriate nucleotide base. And yan, tapos na yung replication. You have now you now have two daughter DNA, no, that are semi-conservative. So yun yung idea. And these are the enzymes used, no? So hindi na natin kwento yung iba dito, but these are their functions, no? So tatlo yung DNA pol 1, 2, 3 natin, no? Okay. So, ayan. So, ayan. So, apparently, DNA pol 3 ang ginagamit to add the bases, the nucleotides. No? DNA pol 1, ito yung nagre-remove ng RNA primer. And DNA pol 2, ito yung nagre-repair ng gaps sa Okazaki fragments. No? Helicase will open the DNA. Ligase, ito yung nag-aayos ng gap. So on so forth. Okay. May video din pala dito, no?
Like these prokaryotic E. coli cells, replicate themselves quickly and efficiently. The aids in the backbones consist of a base pairing in the molecule.
Siguro maybe we can watch this
like
also. Naririnig nyo naman, no? No, let's watch this na lang para may review tayo ulit, no? Before we proceed with the next process. Ayan, we're done with the first part, the replication. Watch na lang natin ulit.
Coli cells replicate themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA before cell division occurs. Prokaryotic cells that reproduce by binary fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make two new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, two identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell, allowing them to function in the same manner. Thus, bacterial populations can grow, increasing the number of individuals in a colony.
Yun lang. Ganun din yun, patulad nun sa kanina. However, medyo may additional info siyang sinabi na. So, mas pinakita niya that yung specific DNA polymerase no, na ginagamit, no, the DNA pol 3, the pol 1, and the pol 2. No. And then pinakita din yun sa SSB, no, yung sinabi ko na kumakabit doon sa nag-split na DNA to stabilize that. Okay? So, prokaryotes pa lang yung na-describe natin, eukaryotes tayo, no? So, ano yung differences? Ano yung similarities, no? Actually, medyo onti lang naman, no? Okay. So, for prokaryotes, we have 5 polymerases. For eukaryotes, ganun din. Okay. So, iba-iba yung function ng polymerase din. DNA pol 1, 2, 3, 4, 5. Ganun din naman sa, sa eukaryotes, alpha, beta, gamma, delta, epsilon. Okay. So, Babasahin natin for prokaryotes, ang polymerase daw ay exonucleases din. Ito yung mga nagre-remove ng RNA primers. For eukaryotes, not all polymerases are exonucleases. No? So there are other enzymes that are used to remove the primers. No? Okay. For prokaryotes, iisa lang yung origin of replication. For eukaryotes, several, no? Kasi remember, yung mga bacteria, maliliit lang yung genetic material nun. For humans, ang dami nun, no? So, yung ating genetic material, yung DNA natin, ay naka-coil yon na super dami. So, imagine nyo kapag isa-isa lang siya, no, mangyari. Ang tagal nun. Tagal nun dumami. So, yan. So, kailangan natin ng several origin of replications for eukaryote. Again, ano yung origin of replication? We cannot really tell where siya exactly. Basta doon nangyayari yung replication, no? Parang Big Bang lang yan, no? Saan yun? Di natin sure. Basta doon nangyayari yung universe. Okay? So, yan. So, for prokaryotic cells, the Okazaki fragments are typically 1,000 to 2,000 residues long. And for eukaryotes, shorter siya. Bakit shorter? Kasi marami naman siyang, marami namang replication processes that happen. Okay. So, ibig sabihin, kahit mas onti yung Okazaki fragments, mabilis pa rin siya. Okay. So, for prokaryotes, now proteins complex to the DNA. Okay, for eukaryotes, histones are complex to the DNA. Histone yung mga circle, no? That helps us to coil the DNA into its chromosome image, no? So, yung DNA natin, kino-coil pa yun ng histones, no? Na kapag nag-coil sila, dun nila na-form yung itsura ng chromosome. Okay? So, kumbaga, coiled talaga yung DNA natin for eukaryotes. But for prokaryotes, hindi naman necessary naka-X pattern yun or naka-Y. Hindi necessary, kaya wala silang histones. Okay? So, yun lang. Also, one difference of eukaryotic and prokaryotic replication is the addition of telomeres. These are sequences that do not code for particular gene. So, kumbaga, ito yung mga safe fails, tawag doon? Parang ito yung mga safety measures na ginagawa ng ating cell, no? Parang there should be data sa ating cell na hindi nagme-make sense, no? Para kunwari, mayroon tayong mutases, no? Kunwari, naputol yung DNA natin. Pag yung telomere yung naapektuhan, edi walang mangyayari, okay? Kasi nga, yung telomere, basically, sila yung mga mga pwede burahin, no? Sila yung mga sequences na wala naman talagang meaning, okay? So, kunwari, meron tayong mga foreign pathogen sa body that directly affects the DNA. Pag tinamaan nila yung telomere, edi no effect sa atin, okay? Parang meron silang safety measures na para hindi talaga super affected tayo just in case na may foreign bodies na pumasok sa ating cells, na. Okay. So, kumbaga yan. Parang okay lang. Yan yung pain niya. Sige, sirain mo yan. Wala namang mangyayari. Parang yun yung idea with the telomere replication. Okay. So now after the replication, edi syempre marami tayong cells. However, to construct the cellular membranes, no? Manggawa yan sa mga proteins, even the enzymes are also made of proteins. To create more of them, no, prior to the separation of the cells, kailangan natin mag-transcribe, no? So hindi lang dapat yung DNA dumadami, kailangan dumadami din yung existing proteins natin. Para yung dalawang cells na mabubuo is complete, no? Hindi pwedeng kulang, no? Yung isa nasa kapitbahay, yung isa nasa isa, no? Hindi gagana yung cell nun. Kailangan ma-replenish din nila yung mga materials nila, no? Ma-duplicate nila. So, to begin that process of reproducing proteins, we have to do transcription. Okay? So, for transcription, medyo complicated to. Okay? So medyo complicated, but I will not go into much deeper details. So let me just summarize this na lang. No? Okay. So let me summarize this. So in transcription, again, this is your DNA. Medyo similar siya with the replication. However, yung splitting ng DNA is not so big, no? Hindi siya ganun ka-grandeur unlike the replication. For transcription, ang mangyayari lang is just a small portion of the DNA is split, no? Hindi siya yung isang buo na hahatiin talaga, no? Ganun kasi sa replication ba, isang buo siyang split. But for transcription, patsi-patsi lang, portions lang. Okay? So, RNA polymerase will split the strand of your new DNAs, no? Split niya 'yan. Pero again, not so big enough, no? Hindi siya katulad ng replication. So, 'yan. So itong ating DNA, mas-split yan into coding strand and the template strand. No? Okay. So the coding strand is the 5' to 3', and the template strand is the 3' to 5' strand. Okay. So ito yung gagalawin ni RNA Pol. No? Pag sinabi kong Pol, polymerase. No? Shortcut namin. So si RNA Pol, kakabit siya sa template strand natin, the 3' to 5' strand. And then, doon niya ikakabit yung mga nucleotide bases for RNA. No? So instead of having T, doon nakakabit yung U. No? So similarities is that ito. So may general steps siya for initiation, elongation, termination. The process of synthesis is the same from 5' to 3' direction. And then adherence to the Watson-Crick base pairing. Ito yung kung ilan yung A, ganun din karami yung T or U. Kung ilan yung C, ganun din karami yung G. No? So it adheres to that rule. No? But the minor differences are this. No? So for RNA synthesis, ribonucleotides are used instead of deoxyribonucleotides. No? So ibig sabihin, the backbone of the RNA should not be deoxyribose. Kasi again, pag sinabi natin deoxyribose, DNA yon. So instead of using deoxysugars, we use oxysugars no? Or ribose sugars. No? Yung may mga OH no? Sa second carbon. First carbon. First pa second? Nakalimutan ko tuloy. Okay. So anyway, basta may OH siya doon. No? And then take note that instead of pairing T, you pair U sa adenine na base. No? Okay. So kailangan uracil instead of thymine because again, we are creating RNA. And also the difference is that, sabi natin kanina, for DNA synthesis, a primer is necessary so that the RNA polymerase can start, no? Para alam niya kung saan siya mag-start, no? Kailangan ng primer. But for RNA, tuloy-tuloy lang. Okay? And then also, only a small portion of the genome will be copied, no? So, onting portion lang, hindi lahat, no? Okay. And then also, no proofreading function. Okay. So, ano itong proofreading na to? 'Di ba sa papansin niyo sa DNA, ang daming polymerase na ginagamit, no? And all those polymerase work no to proofread then yung base materials natin. Okay? So, the reason for using proofreading enzymes such as the exonucleases, no? Ang purpose nila is to prevent yung mutation sa ating strand, no? So, given na magkaroon ng mutation doon, mali na yung chromosome mo. It will result to abnormality na. Okay? So, yun. Kapag nag-take kayo ng full biochemistry course, which is kapag chemistry kami or biochemistry, 5 units to na 2 SEM yung biochem namin. Yan, ma-discuss ko pa doon in details yung proofreading. Pero lang tayo, overview lang. No? 3 units lang tayo. Okay? So, yun. So, for RNA, walang proofreading. Basta ma-photocopy, okay na yun. Okay. So, ganun. So in transcription of prokaryotes, no, sa prokaryotes ulit inaaral kasi nga madali sila, no. So kailangan natin ng RNA polymerase, no, and this is the enzyme that helps us to produce the RNA single strand, no. So this enzyme is a multi-subunit structure. Ibig sabihin ito sa quaternary structure, no. So you have the alpha 2, omega, beta, beta prime, sigma subunit, no. Ayan yung buong composition niya. Okay? So, in short, dalawang components lang yan. Okay? Pero ito yung name niya talaga, no? Pero don't worry, di ko naman kabisado yan, eh. And hindi siya necessary kabisaduhin. Okay? So, yan. So, para yung sandwich, no? Para yung burger, no? Tapos iipitin niya doon yung codings, yung template strand ng DNA. Okay? So, dun sa ating DNA, di ba? The reason bakit hindi na kailangan niya ng primer is embedded na kasi sa DNA yung ano natin, promoters, no? Instead of primer, promoters na yung nakalagay sa DNA. So baling gagawin ni gagawin ni RNA polymerase, 'di ba? May strand ka ng DNA. Hahanapin niya yung promoters. Okay, hahanapin niya yung promoters. Ano yung promoters na yon? Typically sila yung tata box, no? Sila yung mga tata box or the Pribnow box, no? So, hanapin niya yun. So, kunwari, ito yung strand ng DNA mo. Hanapin niya yan, no? Basahin niya, saan kaya dito yung Pribnow box? Ay, pag nakita niya, ay, ito Pribnow box. So, dito na siya 10 nucleotide bases after that, dun siya mag-start. Okay? So, ganun. Parang ano siya, nag-find siya. Asan kaya yung yung promoter ko? Pag nakita niya yung promoter na Pribnow box or the Tata box, yun. Ayun, dun siya mag-start, no? Okay. So, ito yung process for the transcription. Okay, so the RNA polymerase will recognize the promoter. Ay, yung promoter box natin ay pwedeng Pribnow or Tata box na. So, hahanapin niya yan sa inyong DNA strand. Hahanapin niya yan. And then, pag nahanap na niya yan, kakabit na siya sa DNA, then split na niya yan. Okay, and then it will start reading the template strand, the 3' to 5' strand. No? So it will start reading that portion of the DNA. So once it starts reading that, then makakabit na yung ating mga nucleotides. No? So kakabit na yung mga nucleotides natin, and then elongation. Yan. So tuloy-tuloy na yan. Then we will have our RNA na. Okay. So ayan, chain initiation. Chain elongation is just the continuation of that process kanina. So ito na yan. So again, to start the transcription process, hahanapin niyo muna yung promoter which is embedded on the DNA itself. Kaya hindi na kailangan ng primer kasi ando na yung promoters. Pag nahanap niya yung bubuksan niya, then babasahin na niya. Okay, from 5' to 3' direction then. And then it will continue elongating, and then after some time mag-stop yan, no? So the termination, meron kaming mga enzyme subunit na inaaral doon. Okay, so once na makita niya sa template stand mo na, uy, may nauulit na to, so ibig sabihin nun, stop na, no? You have to stop making the RNA na. So may makikita siya mga row independent na sequences. Okay. So, pag nakita niyo yun sa DNA, stop na siya. Okay. Ayan. So yung termination site, yun yung mga portions of the DNA that will tell the RNA polymerase to stop producing mRNAs, no. So yan. Then once na tumama na ito yung termination site. Sequence yan ng DNA. So pag tumama na yun sa RNA pol, sabi niya, "Oy, stop na." Ayan, hinto na siya. Okay. So tapos na yung transcription natin. For eukaryotes, medyo similar yung process, no? There are specific enzymes used in eukaryotes such as RNA pol 1, 2, 3. Okay? And yan yung kanilang mga functions, no? Okay? So I will not go into much details na. So, andito yung tatap box na sinabi ko. Ayan, ito yung tatap box. Hanapin niyo yung tatap box, pati yung pribnode box. Then, once na mahanap na yon, makapag-start na siya. Okay? Okay. So, ito yung mga post-transcriptional methods na lang. Ito yung mga additional steps that are taken by the cell, no, to make sure that yung mapoproduce niyang RNA is okay na ilabas papuntang ribosome. Okay? Kasi from nucleus pa rin to eh. Papadala niyo yun sa ribosome. So, after transcribing the DNA, i-check niyo muna whether okay na yung aking mRNA. So, may mga iilang process yan. For example, splicing. So, when we say splicing, it removes the introns and connects the extrons. Ano yun? Basically, yung mRNA strand, pinuputol niya, tapos pinagdudugtong niya ulit. No? So, ganun ginagawa ng splicing. Putol, then kakabit. No? Just to make sure na correct yung kanyang na form. Okay. So, parang ang weird, no? Kaya nila gawin yan in very quick fashion, no? Wala lang, nakaka-amaze yung body natin, no? Parang we are really made as, parang katawan natin is a big factory, no? So, yan, automatic gagawin ng ating mga, ng ating mga enzyme doon, hahanapin niya yung ano yung mga introns, yung mga puputulin, no? And yung exons yung mga kailangan pagdugtongin. Okay? So, yan. So anyway, so yun yung splicing, no? Putol-putol, then kabit, no? Then sumunod is the 5' cap, no? So yung 5' cap, ito yung mga modification sa 5' end ng ating mRNA so that whenever na lumabas siya sa nucleus, pag nandun na siya sa pag na siya ay nasa cytoplasm na, sa cytoplasm kasi medyo weird yung nangyayari dyan, no? Parang yan eh, sabihin na natin parang recto yan eh. Okay? Hindi mo alam kung mananakawang ka o hindi eh. So parang yung mRNA, siya yung baby na lalabas sa recto. And hindi niya alam kung ano mangyayari sa kanya. So, ang ginagawa ng mRNA or ng RNA, bago siya palabasin sa nucleus, no, is minomodify siya. Yung splicing, ito, wag mo to dalhin, bla bla bla. Ito yung dadalhin mo sa ribosome. Yung 5' cap, ito naman yung mga modification at the end of the 5' in which pinapalit niya yung dun sa guanyl group sa 5', ginagawa niyang methyl group, no? So, kino-convert niya lang into methylated form. So, as a result, hindi siya makakapagkaroon ng chemical reaction with other components in the cell, the cytosol, no? Okay? So, hindi siya magkakaroon ng other chemical reactions when that residue is converted to methyl group, no? So, ganun lang. Papalitan niya lang, no? And then additional step, no, bago siya palabasin is the addition of the poly-A tail, no? Or the poly-A tail or polyadenylate tail. Ito yung nakikita nyo sa memes sa Facebook na ayan. So, yung puro A sa dulo. Okay. So, dinudugtong niya yan sa dulo. No? So, kung yung 5' papalitan niya, gagawin niyang methyl group yon, so it will not react with other chemicals out there. Yung dulo naman, papalitan niya ng poly-A, or the polyadenylate tail. Okay. So, itong protection na to, this ensures that our mRNA will not react with other molecules outside the nucleus, no, sa cytoplasm. Kasi ang ribosome medyo malayo from the nucleus, no. Tatawid ka pa ng Golgi bodies, no. Dadaan ka pa ng smooth ER, rough ER, then mamaya ayun, ribosome na. Okay? So yan, medyo malayo kasi kaya kailangan niya ng protection, no. Okay. So, itong purpose ng poly-A tail, kunwari may mga chemicals sa cytosol natin na sisirain yung mRNA, kunwari kakagatin siya. At least kapag naputol yung poly-A tail, wala mangyayari. Preserve pa rin yung message, no. Okay lang kagatin yung poly-A tail ng enzymes na nasa labas ng cytoplasm. Ayan. So, medyo harsh yung environment sa cytoplasm, no? Kala mo okay lang no? Pero imagine mo yung mRNA, it has to undergo all of this stuff, no? Para lang ma-translate siya into proteins, no? Ibig sabihin, ang cytoplasm ay isang recto. No? Kailangan mo mag-ingat. No? So, kailangan mag-disguise ka pa. Lagyan mo pa yung sarili mo ng 5' cap, no? The methyl cap. And kailangan lagyan mo pa yung sarili mo ng poly-A tail. No? So that hindi ka masisira doon. Hindi ka aawayin ng ibang molecules. No? Okay. So, yan yung idea. No? So, yan. So, once na siya ay makapunta na sa ating so, ito pala yung example ng sinabi ko. So, ginagawa dito yung mga introns, pinuputol-putol, splicing tawag doon. Yung 5' ginagawang methyl group. Okay? And then yung dulo, dinudugtungan ng A. No? Usually 100 to 200 100 pieces or 200 pieces no, ng A yung ilalagay niya doon. So once you have this final mRNA, you have your thing called mature mRNA. Mature, ibig sabihin pwede na siya lumabas. Okay. Pero kapag ganito pa siya, bawal pa siya lumabas. No? Kailangan maging mature muna siya so that it can go outside. Okay? And then yan, no? Magta-travel siya from nucleus, may butas doon sa nucleus, lalabas siya. Then it will proceed to the ribosome where the translation will happen. Okay? So in the translation process, dito na nako-convert yung ating sequences into proteins, no? Okay. So let's talk about genes, no? Okay. So when you say genes, ito yung parts ng chromosome natin that translates into a code of polypeptide, no? Okay. Parang ito yung code na na pwede i-translate into proteins, no? So yung ating DNA or ating chromosomes, no? Yung mRNA, yung na ipoproduce nun, pagpunta yun sa ribosome, i-convert yun into proteins, no? Yung portion ng DNA kung saan niya nakuha yung mRNA, tawag doon ay gene, okay? So, ang characteristic ng genetic codes natin ay ito. Number 1, it is triplet. Ibig sabihin, 3 nucleotide bases are necessary to convert that into one single amino acid. Number 2, non-overlapping. The reading of codons do not overlap. Ibig sabihin yan, kunwari you have your codon A, T, A, C, G, A, G, C. Pag binasa niya yan, ito muna babasahin ko, ayan, then followed by this, followed by that. So pag sinabi kasing overlapping, ito muna, tapos ito, tapos ayun, hindi ganun yung nangyayari. So ang ginagawa natin sa ribosome is that babasahin niyo yung 3, tapos susunod yung 3, then susunod yung 3. Hindi yung katulad nito na ito, tapos ayun, tapos yun, so on and so forth. So non-overlapping. Number 3, comma-less. Ibig sabihin, sunod-sunod na letter lang yan. So itong comma-less, ano yan? Sa way ng pagsulat natin. No? Tuloy-tuloy lang yung letter niyan. Okay. Number 4, degenerate. No? So when we say degenerate, no, we have multiple variation that correspond to one amino acid. No? So the purpose of degeneracy of our codons is that to prevent the mutations in our proteins. Okay? So yan. So kaya siya degenerate para kahit magbago yung isang codon, okay lang. It will still be translated to the same amino acid. Okay. So kumbaga ito yung mga fail-safe measure niya, no. Okay. So, para hindi palyado agad yung protein na mabubuo, kailangan may mga extra measure siya, no. May mga siya, redundancy. Okay, such as the degeneracy. Unambiguous, ibig sabihin yan, only one amino acid will correspond to one codon. Okay? So, ibig sabihin, yan. So, itong codon na to, this will be translated to one specific amino acid and hindi sa iba. Itong codon na to, ibang amino acid mo po-produce niyan. Okay? And then, lastly, ito yung important, universal. That's why pwede interchangeable yung gawin nating study for eukaryotes and prokaryotes. Universal because kung ano yung codes for the prokaryotes can be used for eukaryotes as well. No? That's why in drug testing field, no, in the field of that research, puro animals yung kanilang tinetest. I mean, puro bacteria yung tinetest nila. Say whatever works for bacteria will work for animals, no, and humans. No? Okay, so yun yung idea doon. And this is the genetic code. Hindi nyo to kakabisaduhin, of course. Ano lang kailangan natin gawin dito? Okay. So, ganito yung process dito. Wala pala dito sa PowerPoint. So when our mRNA goes to mRNA, sulat ko na lang. When our mRNA goes to the ribosome, 'di ba yung mature messenger RNA natin, pag pumunta na siya sa ribosome, ang gagawin ng ribosome is hahanapin niya yung start codon. The start codon is AUG, no? Kunwari, we have this sequence. Okay. No, kunwari, we have this sequence. Ito yung 5', tas ito na yung ito na yung 3' end ng ating mRNA. So, ang gagawin niya, hindi agad siya mag-start dun sa first sequence, no? Hindi siya agad dun mag-start sa C. Hahanapin niya muna yung start codon, no? Hahanapin yung start codon, which is the AUG. Ang mnemonic namin dito is, kailan ba nag-start yung classes? August. So, sa August, ito yung start ng classes ng academic year. Okay. So, the start codon is the AUG, the August. Okay. So, hahanapin niya muna. So, from 5' to 3', hahanapin niya yung August, no? So, syempre, itong tatlo, hindi dito siya mag-start, no? Hindi siya dyan mag-start. Kasi CAU yan eh. Hanapin niya yung AUG. Ayun, AUG, August. So, once na makita na niya yan, edi tatlo-tatlo na yung gagawin mo dyan. Okay? So, ayan, start na yan. Start na to ng protein. So, dito na yung N-terminal ng protein. Okay? So, ayan. So, dito na yung N-terminal, August. Okay? So, ayan. So, this is the N-terminal. So, apparently, yung AUG, it translates into the amino acid na methionine. Okay? Continue tayo. So, methionine yung first sequence natin ng ating protein from our mRNA. Sunod, AUG. Ano susunod na sa sequence? Hindi yung UGU. Kasi sabi nga natin, hindi siya nag-overlap. Okay? So, kung AUG yung sumunod, kailangan yung next 3 yung susunod. Hindi yung katabi, no? Kung AUG, hindi UG yung sunod. Instead, UAU, no? Ito yung susunod. Okay? So, ganun. So, after 3, yung next 3 susunod. Hindi overlapping, ha? What is UAU? Hanapin mo lang dito, UAU. U-A-S-U. Ayun, UAU. That's tyrosine. The next 3, codon will give us UGG. UG, and then the G, tryptophan. And the UGA, stop. Ano meaning ng stop? Ito, tapos na. Carboxyl group na yun. Okay? So, yun. Ang translation ng ating protein will always end with the end codon. Okay? Ang end codons natin ay 3, no? Pwedeng UAA. Ayusin ko siya sa mnemonic namin. UAG, UGA, UAA, no. Ito yung stop codon, no. So pag nakita niyo yung stop codon na UGA, carboxyl end na 'yan. Okay. So yan, so this is your proteins na. Okay. Ano yung mnemonic namin for the end codon na ganito? Wag uga uwa, no. Ibig sabihin, wag iuga kasi iiyak, no. Wag uga iwa. Uwa. So, ang English translation dito ng prof namin is, don't rock the cradle because the baby will cry. Huwag iuga ang cradle para hindi uwa ang baby, no? Hindi siya iiyak, uwa, no? So, yun lang. So, very helpful yan until now, naaalala ko yun. Yung ibang topic sa biochem, di na, pero ito, naaalala ko pa rin. So, yun lang. So, yan. So, mnemonic, AUG, dun mag-start, and always the starting protein, the starting amino acid will be the methionine. And then, once it reaches the wag uga uwa codon, then stop na yon, carboxyl group na yon. Okay? Let's practice pa. Try pa tayo. Nakakatuwa, no? Kasi yung na-discuss nating proteins, dito lang din pala siya babalik, no? Very related yung topics natin. I will give you a sequence from 5' to 3'. Then, try natin siya i-translate, ha? Manghuhula na lang ako ng letters. Ayan. So, I have my poly A tail at the end. Okay. So, ayan. So, I want you to try to translate that mRNA into peptide chain. Okay? So, I want you to translate that into proteins. Sige, I'll give you 5 minutes to do that. Sige. Dagdagan ko pa para mas magamit natin. Ayan, so you have to genetic code to convert into proteins. So paki-comment na lang sa chat box, no. Ayan. So always ang clue ko sa inyo is to find the August. Hanapin niyo yung start codon. And the start codon will always be translated as methionine. Okay? And then the others will follow na lang. Okay?
Sir, ano nga po ulit, sir, yung nilalagay before ng mag-AUG?
Yung
NH3 plus?
NH3. The N-terminal of the protein. By the way, naalala ko na, no. For deoxyribose, it should be the two sugar, no? The second carbon dun dapat. Okay. Okay, so yan. We have answer from Nicart for the first one. Okay, so may isa pang sasagutan si Rika. So while waiting for the second answer, maybe we can check the first one. Okay, so let's check kung sino yung first, ano yung tamang sagot. Kasi iba kay Nicart, iba kay Rika. Okay, so let's decode this. Again, always look for the August or the AUG because that is the start codon, which translates to methionine. So itong GUA, hindi yan. UAU, hindi rin yan. AUG, ayun. So you will start here. Followed by the next three, the next three, the next three, the next three, and stop ka na dyan. Kasi you have your uwa, no? Always end sa uwag, uga, uwa. No? So magigets nyo na yung yung meme sa Facebook, no? Instead of using stoplight, nakalagay doon, uwag, uga, uwa. No? Stop. Yan. I will stop here. This is the C terminal of our amino acid. Okay. So as I told you, the AUG is the methionine. So you have your NH3+. This is your methionine, followed by the UCU. Ito, letter U, letter C, hanapin yung U, ayan. UCU, that's serine. AGG, AG, tapos G, ayan. AGG is arginine. AGG din ilalagay ko. Arginine. Ayan yung may yan, ito yung source ng urea sa body, the arginine. You have the GCC, G, C, then C, GCC. Ayan, that's alanine. Okay. GCC followed by CGC, arginine. Ayan. So, dito lang na ano tayo. So, this is ano daw? Wala tuloy ako. CGC, this is arginine again. And then, you have your UAA, which is the stop codon. So, you write COOH. That's your amino acid, no? Metzer, Arg, Arg, stop. Ayan. Pwede nyo na isulat yung stop na, just the COOH. Yan. Okay, so yan. So, okay kay Rika. Si Nicart, siguro na nakakalito din kasi yung letters, no? Dikit-dikit eh. Okay. So, kaya ang gagawin ko kapag kapag nag-exam kayo, may ganito, ano na lang, wala nang time, no? Basta sagutan nyo na lang. Or siguro kung mag-time man ako, medyo matagal, mga 2 hours. Ayan. Kaso 25 items lang yun eh. Siguro 1.5 hour. Para hindi kayo malito sa pag-highlight. Okay? So, yan. So, this is the amino acid sequence. About this one, we have the CAA. Hanapin yung August sa 5' end. Ayan. So, this is the August. And then, tigta-tatlo. Oops, stop ka na dyan. Uga na yan. Wag, uga, uwa na. So, you stop pag nakita mo na yung uga na. So, that's COOH. So, the August is the methionine. The ugu, that's cysteine. So, the second amino acid sequence is the met and the cis, no? Ayan, so both got it correct. And kahit COOH na ilagay nyo dun sa dulo. Okay? So, ganun mangyayari. So, imagine nyo, ano kaya possible na lumabas sa final exam? So, pwedeng lumabas sa final exam is I will give you the leading strand of, I will give you the template strand of the DNA, and later on you will convert that to protein. Sasabihin ko na rin saan kayo mag-start mag-encode ng mRNA. Ilalagay ko na doon. Di ko na ipapagawa yung Pribno box, yung Tata box, hindi na. No? Kasi that's too complicated na. Okay. Sasabihin ko, start kayo mag-encode dito. You should be able to find the correct amino acid sequence for your resulting protein. Okay. So, ito na lang mga additional steps, no? So, before you produce your proteins, no? Ito muna yung nangyayari. The activation of your amino acid. Then this activated amino acid will be added to your growing polymer chain until the termination happens. No? By the way, if asked, no, ano yung tumutulong sa production ng proteins from the mRNA? Ang ginagamit natin doon is the tRNAs, no? Or the transfer RNAs. So these are also RNA molecules that help in making the peptide sequence. Okay. So this is the tRNA. Obvious naman yung letter T siya. So it is composed of many parts. So you have your acceptor stem, you have the D loop, and then you have your T C loop, and then you have your anticodon. Okay. So asan dito tumatapat yung ating mRNA? Dito sa anticodon. Okay. So, dyan siya tatapat na. Tapos, kapag tumapat dyan yung codon natin, sasabihin niya sa acceptor RNA, kuha ka methionine. Tapos gagawin yan, pag nandyan na yung methionine, yung activated methionine for bonding, ipapasa niya yon papunta sa baba. Okay? So, mabubuo na yung ating methionine portion. Then the next sequence susunod. Kunwari, you have your UGU. Ano yung translation ng UGU? Cysteine. Sasabihin niya, may UGU dito sa baba. Maglabas ka ng cysteine. So gagawin ni acceptor stem, kukuha siya ng UGU sa paligid. Ipapasa niya yon sa baba. No? So, ganun gagawin niya. Papasa-pasa lang ng amino acid sequence. No? Until you create your protein from your mRNA. Okay? So these are the parts of the tRNA, the transfer RNA, na tumutulong in creating the protein. No? This is found in the ribosomes. So you have your T size C, the D arm, the anticodon, and the acceptor arm. And ito yung kanilang mga purposes. In prokaryotic cell, of course, medyo complex lalo. So instead of just having the tRNA, we have this thing called the ribosome. Okay? So may ribosome na tayo. So the ribosome is composed of 3 groups, no? The A, P, and E. Tawag dyan ay A, pero pabaliktad. The EPA. So dito papasok yung mRNA natin. So dito papasok yung 5' to 3'. And then, ang mangyayari dyan, the A group will be the amino acyl group. No? So, dito ikakabit yung amino acid. Nababasa niya, AUG. Ayan, lalagay niya dyan sa baba, AUG. Then, pagdating dito, syempre, magbabasa ng panibagong sequence yan. Cysteine. Ibababa niya yung cysteine. So, by the time na tumapat yung ating amino acids dito sa P, which is the peptidyl site, dito na form yung peptide bonds. No? So, dito nangyayari yung hydrolysis ng bond, no, to form the peptide bond. And the E is the exit side. So, dun nalalabas yung ating protein. No? And then, once na lumabas yung protein, remember our discussion in proteins, instantly it will fold to its final tertiary structure or the final quaternary structure if it's multi-subunit. Okay? So pagkalabas dyan sa E site ng ribosome, magpo-fold na yan bigla to form the final structure of the protein. Okay? As you can see, our topic is very integrated to one another. No? So yan. Ay, ang dami nangyari. Anyway, so ayan. So this is the elongation. Ayan, no? 5' to 3' yung pasok ng ating Yan, dito nadudugtong yung ating mga amino acid on the P site. No? So sa P site na bubuo yung peptide chain. Kaya nga letter P siya. Okay. And then kapag tumapat na yung AUG, yung wag uga uwa, no? Pag tumapat na yan, madi-disassemble na uli yan lahat. Okay? Madi-disassemble siya. Okay? So take note that in eukaryotic translation, may minor differences tayo. We have the mature mRNA has a 5' cap and a poly A tail. Okay? And then second one, the Kozak sequence precedes the AUG start codon. So ibig sabihin, before you see the AUG, bago ka mag-start mismo sa AUG, ay makikita ka muna ng ACCA UGG. So, hindi ka dito mag-start, instead dito. Okay. So, this thing is called the Kozak sequence. So, sasabihin ko naman sa finals, no, kung if you have to consider the Kozak sequence or not. No. Kapag hindi ko sinabi na wag na i-consider yung Kozak sequence, hindi dito ka mag-start. No. Pero kapag sinabi ko, look for the Kozak sequence, ibig sabihin nun, pag nakita mo to, wag ka mag-start dito sa AUG, rather dun sa katabi. Okay. So imagine the body is too complicated to handle all of this stuff, no? Kaya it is very it's very parang mapapaisip ka talaga, no? Imagine mo if yung molecules lang talaga ba yung driving force, no? Starting our first topic, no? So yung lang ba yan, yung mga molecules, yung mga atoms lang ba yan nagsama-sama to form the simple organic molecules, then later on biomolecules, then nagkaroon ng living species. Ganun lang ba talaga yung essence ng life, or there is something that created us, no? Parang kasi makikita ko ang daming nangyayari, no? This is too complicated. As you can see, replication pa lang, ang dami nang nangyayari, no? So, yun, no? So, this will make us question whether nag-exist lang ba talaga tayo because of the molecules or someone created us from nothing, no? So, yan, no? Pero nonetheless, no, uh, yung ating yung existence natin is a living proof, no, that really chemistry is chemistry can help us understand everything, no. Ayan. So ito yung patunay na na with the help of chemistry we can understand life, no, and by understanding life we can make other lives better din, basta yun lang. Okay. So, ano yung major takeaways nito? Other than the lesson we discussed, no? Other than that, ang gusto ko gawin nyo is that pag umuwi kayo, nasa bahay nga pala kayo. Pagkatapos nito, bumili na kayo ng vitamins or multivitamins with zinc. Because remember, yung zinc fork, no? Once it is stabilized, no? Mas mabilis yung replication process. Sabihin, mas healthy yung ating cells sa katawan. Ikalawa, ang gusto ko sabihin is that continue to look for the current trends in molecular biology, no? So continue to look for for updates, no? Kasi lately with the technology called the mRNA vaccine, no? So yung mRNA vaccine yun, dumadaan yun sa ribosome to create the protein of the COVID-19. Pag nabuo na yung protein ng COVID-19, mamumuo na agad yun as crown. Galing, no? Okay, so anyway, so I want you to be very updated na sa mga ganung topic, no? Although biology siya or chemistry siya, yes, no. Kung hindi nyo bet yung chem or bio, maganda pa rin yung updated tayo, no, kasi we will understand life more, no, kapag ganun. Ano pa yung kailangan ko i-remind sa inyo? To summarize everything, let's watch this short video clip. Nakita ko lang din to sa YouTube kanina. So this is the summary of everything we discussed, no, in just a few seconds.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, A transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Ayan na. So as you can see, very complex yung nangyayari sa buhay. So I hope this topic made us aware of the molecular processes in the body, and hopefully makatulong din to allow us to have better understanding of what's happening around us, especially during the times of COVID. Now you know the meaning of RT-PCR. Okay, so as your assignment, as your quiz, no? So wala muna tayong multiple choice quiz, rather ilalagay ko siya sa finals, okay? Pero ito yung mangyayari, no? So I think na-upload ko na siya sa YouTube. Ayun, na-upload ko na. Hindi, sa Google Drive ko. So mag-a-assign ako ng assignment na gawin ko na siya ngayon kasi kaka-upload ko lang sa Google Drive. So I want you to watch this, no? Teka lang. Upload error. Upload error pa pala. Anyway, so gawan ko to ng paraan, no? So I will upload this, no? You can download it actually. Malakas ba internet nyo dyan? Unlinet naman ba kayo?
Yes po.
Unlimited na internet nyo, guys?
Yes po.
Okay. So kung yes naman, so siguro magbibigay na lang ako sa inyo ng download link. Upload ko na lang to sa aking OneDrive, no? So please download the movie Erin Brockovich, no? And that is a very inspiring movie. Medyo comedic siya in a sense, pero if you look at it at a different perspective, no, mas ma-appreciate mo yung yung service ni Erin Brockovich to the people, given na related to sa biochem, no. So si Erin Brockovich kasi, to give you an insight, siya ay isang normal na babae lang, unemployed, walang trabaho. Tapos, she was very concerned with other people sa kanyang paligid kasi nagkakasakit sila. Iba nagkaka-cancer. Tapos, ang naging sala rin doon is yung company. Basta yun, panoorin nyo na lang. Na. So, again, gusto ko i-relate nyo yung movie. No? Mamaya, upload ko sa Canvas yung link kapag na-upload ka na. Ha? So, gusto ko i-relate nyo to with several issues. For example, sa Flint, Michigan, yung water contamination doon, which is relatively similar with Erin Brockovich's story. Kung meron sa Pilipinas kayong makitang ganyan, look for articles then, relate that. Okay, so I will upload a rubric, no, kung paano kayo mag-gradean for for this quiz na. Okay? Ano pa ba kailangan ko sabihin? Ayan, no. So next meeting, wala na tayong synchronous session. So if not Monday, baka Tuesday, doon ako makapag-upload ng materials for next week, no. So pagsasamahin ko na yung topic natin sa carbohydrates and sa metabolic pathways, no. Magkasunod lang naman talaga yan. Okay, so for the carbohydrates, we will discuss sugars. Okay. We will discuss sugars, ano yung role ng sugar, saan gawa yung sugar, ano yung structure ng sugar, how to convert this linear sugar molecule into cyclic form. Please recall your org chem, ha, kasi mag-chair confirmation tayo. Nagganon tayo, di ba? Alam ko slight lang yung ganun natin. Anyway, so pero majority of the structure for sugar are in Hayward projections naman. Anyway, so didiscuss natin yung isomerisms as well, the types of sugar. Hindi kailangan kabisaduhin yon. Marami yun eh. The types of sugar, and then how some specific examples of sugar, and the glycoconjugates in storage form ng ating sugar, and the reactions of sugars. Then after that, we will proceed with the last topic, which is about the metabolic pathways. Marami to, no? Tatlo to. We have the glycolysis, Krebs cycle, and the electron transport chain. Okay. However, di ko na i-discuss to lahat, no? Pero ito yung nangyayari. This is the energy pathways for our body, no? Kaya tayo nabubuhay. So we start with glycolysis. We produce the pyruvate. Then they are converted to acetyl-CoA. Then the acetyl-CoA will go to citric acid cycle. They'll be converted to an NADH and FADH2. Nicotinamine, adenine, di something. Dihydrogen something. Okay. So yan. Then this NADH, FADH2 will be used in electron transport chain, which is very highly energetic. Pero ang i-discuss ko lang primary dito is the glycolysis, no? So just the basics, no? Yung Krebs cycle and the electron transport chain, pwedeng na lang 'yon. Mabilisan na lang, ayan. OXPHOS ang tawag sa electron transport chain, oxidative phosphorylation. Mabilisan na lang yan, kasi super daming nangyayari dyan. Kasi nga, kung kami BS Chem, 2 SEM namin to inaral. So sa inyo na isang SEM lang, mabilisan lang tayo. Okay? So ayan, so that will conclude our semester. By next week, tapos na yung ating semester. And of course, as always, it's been a pleasure to teach you guys, no? Hopefully, kayong mga future educator, in the future, maging rin kayo, maging science major, no? Especially chemistry majors, no? And medyo relatively important na yung chemistry teachers sa Philippines, no? Kasi lumalawak na yung ating chemistry program sa Pilipinas, no? Dumadami na yung mga nag-aspire maging chemist, no? So yan, dati kasi nung ako BS Chem, kami parati sa PUP yung last na ine-enrollan, no? Kasi ang gusto nila engineering, gusto nila accountancy. Pero recently, no, nagiging first choice na ng students ang chemistry. Okay? So I hope yung chemistry na tinuturo sa inyo maging inspiration kahit paano to pursue chemical sciences, no? And masters in the future, no? Ayan, basta magiging teacher din kayo in the future, no? Ayan, so yan. So next semester, analytical chemistry kayo, no? So that is the subject I am teaching now sa MedTech. Hindi ko alam kung sino magiging teacher nyo dito, pero bigyan ko na kayo ng insight sa Anachem. Anachem is a very mathematical topic, no? So ang daming solving dito sa Anachem. So if ever di ako maging teacher nyo, you can ask me for materials, no? I can give you some materials for Anachem, no? And then also in my YouTube channel, may mga Anachem videos ako doon that you can watch in advance, no? Okay? So yan, as always, it's been a pleasure to teach future teachers, no? So I'll see you again in the future, no? So hopefully pag nakita ko na kayo, mga licensed teachers na kayo, na. Okay. So yan, sama-sama tayo mag-improve ng knowledge sa Pilipinas, no? By being effective teachers, no? Okay, so with that, medyo nagpapaalam na ako kasi last synchronous na natin, no? Okay, so with that, no, I'll see you again somewhere, I don't know kung saan, no? Kita-kita na lang in the future, okay? So all the best and good luck to your future endeavors, okay? So hindi pa tayo tapos sa SEM, may gagawin pa, pero goodbye na ako kasi asynchronous na tayo nun. Okay? So thank you very much and good luck mga future teachers. Bye-bye and thank you.
Thank you po, sir.